Now you're going to learn structure of gametes, a developmental biology portion. Sperm was discovered in the 1670s, but their role in fertilization was not discovered until the mid 80s. It was only in the 1840, after Albert von Kueliger described the formation of sperm from cells in adult testes, that fertilization research could really begin. Even so, von Kueliger denied that there was any physical contact between sperm and egg. He believed that the sperm excited the egg to develop in much the same way a magnet communicates its presence to iron. The first description of fertilization was published in 1847 by Carl Ernst von Baer, who showed the union of egg and sperm in sea urchins and tunicates. He described the fertilization envelope, the migration of the sperm nucleus to the center of the egg, and the consequent early cell divisions of development. Sperm anatomy. Each sperm cell consists of a diploid nucleus, a propulsion system to move the nucleus, and a sac of enzymes that enable the nucleus to enter the egg. In most species, almost all of the cell's cytoplasm is eliminated during sperm maturation, leaving only certain organelles that are modified for spermatic function. So, each sperm cell consists of a haploid nucleus, a propulsion system to move the nucleus, and a sac of enzymes that enable the nucleus to enter the egg. In most species, almost all of the cell cytoplasm is eliminated during sperm maturation, leaving only certain organelles that are modified for spermatic function. During the course of maturation, the sperm's haploid nucleus becomes very streamlined and its DNA becomes tightly compressed. In front or to the side of this compressed haploid nucleus lies the acrosomal vesicle or acrosome. The acrosome is derived from the cell's Golgi apparatus and contains enzymes that digest proteins and complex sugars. So, enzymes stored in the acrosome can digest a pass through the outer coverings of the egg. In many species, a region of actin proteins lies between the sperm nucleus and the acrosomal vesicle. These proteins are used to extend a finger-like acrosomal process from the sperm during the early stages of fertilization. The sea urchins and numerous other species, recognition between sperm and egg involves molecules on the membrane of the acrosomal process. So together, acrosome and nucleus constitute the sperm head. The means by which sperm are propelled vary according to how the species has adapted to environmental conditions. In most species, an individual sperm is able to travel by whipping its flagella. The major motor portion of the flagellum is the axonium, a structure formed by microtubules emanating from one of the two central microtubules surrounded by a row of 9 doublet microtubules. These microtubules are made exclusively of the dimeric protein tubule. The outer centriole is also important as it will enter the egg to establish the mitotic spindle of first cleavage. Nucleus, Golgi apparatus, mitochondria, centriole, acrosomal vesicle and granule, flagellum, centriole, flagellum, Golgi, remnant, mitochondria, then acrosomal vesicle, cell membrane, nucleus, haploid nucleus, mitochondria, centriole, axonium, sperm head, midpiece, tail and end piece. Modification of a germ cell to form a mammalian sperm. The centriole duplicate using one centriole to organize a long flagellum, but what will be the posterior end of the sperm? The other centriole will enter the egg at fertilization. The Golgi apparatus forms the acrosomal vesicle at the future anterior end. Mitochondria collect around the flagellum near the base of the haploid nucleus and become incorporated into the midpiece or the neck of the sperm. The remaining cytoplasm is jettisoned and the nucleus condenses. The size of the mature sperm has been enlarged relative to the other stages. Mature bull sperm. The DNA is stained blue, mitochondria are stained green, and the tubulin of the flagellum is stained red. Although tubulin is the basis for the structure of the flagellum, other proteins are also critical for flagella function. The force for sperm propulsion is provided by dynein, a protein attached to the microtubules. Dynein is an ATPase, an enzyme that hydrolyzes ATP, converting the released chemical energy into mechanical energy that propels the sperm. This energy allows the active sliding of the outer doublet of microtubules, causing the flagellum to bend. So the ATP needed to move the flagellum and propel the sperm comes from rings of mitochondria located in the midpiece of the sperm. In many species, and notably mammals, a layer of dense fibers has interposed itself between the mitochondrial sheath and the cell membrane. This fiber layer stiffens the sperm tip because the thickness of this layer decreases towards the tip. The fibers probably increase the efficiency of forward movements by preventing the sperm head from being whipped around too suddenly. Thus, the sperm cell has undergone extensive modifications for the transport of its nucleus to the egg. An egg, cytoplasm and nucleus, all the material necessary to begin growth and development must be stored in the egg or ovum. Whereas the sperm eliminates most of its cytoplasm as it matures, the developing egg, called the oocyte before it reaches the stage of meiosis at which it is fertilized, not only conserves the material it has, but actively accumulates more. The meiotic divisions that form the oocyte conserve its cytoplasm rather than giving half of it away. At the same time, the oocyte either synthesizes or absorbs proteins such as yolk that act as food reservoirs for developing embryo. Birds' eggs are enormous single cells swollen with accumulated yolk. Even eggs with relatively sparse yolk are with large compared with sperm. The volume of sea urchin egg is about 200 picoliters, more than 10,000 times the volume of sea urchin sperm. So even though sperm and egg have equal haploid nuclear components, the egg accumulates a remarkable cytoplasmic storehouse during its maturation. The cytoplasmic trove include the following: female pronucleus, egg cell membrane, vitelline envelope, jelly coat. Structure of a sea urchin egg at fertilization. Sperm can be seen in the jelly coat and attached to the vitelline envelope. The female pronucleus is apparent within the egg cytoplasm. Nutritive proteins. The early embryonic cells must have a supply of energy and amino acids. In many species, it is accomplished by accumulating yolk proteins in the egg. Many of these yolk proteins are made in other organs like liver, fat bodies, and travel through the maternal blood to the oocyte. And ribosomes and tRNA. The early embryo must make many of its own structural proteins and enzymes, and in some species there is a burst of protein synthesis soon after fertilization. This rapid protein synthesis is accomplished by those ribosomes and tRNAs that exist in the egg before fertilization. The developing egg has special mechanisms for synthesizing ribosomes. Certain amphibian oocytes produce as many as 10 ribosomes during their meiotic prophase. And messenger RNA. The oocyte accumulate not only proteins but also mRNAs that encode proteins for the early stages of development. It is estimated that sea urchin eggs contain thousands of different types of mRNA that remain repressed until after fertilization. Then morphogenetic factors. Molecules that direct the differentiation of cells into certain cell types are present in the cell. These include transcription factors and Varrockine factors. In many species, these factors are localized in different regions of the egg and become segregated into different cells during cleavage. And protective chemicals are also there. The embryo cannot run away from the predators or move to a safer environment, so it must be equipped to deal with threats. Many eggs contain ultraviolet filters and DNA repair enzymes that protect them from sunlight, and some eggs contain molecules that potential predators find distasteful. The yolk of bird eggs contain antibodies that protect the embryo against microbes. Within the enormous volume of egg cytoplasm resides a large nucleus. In a few species, such as sea urchins, this female pronucleus is already haploid at the time of fertilization. In other species, including many worms and insects, the egg nucleus is still diploid. The sperm enters before the egg's meiotic divisions are completed. In this species, the final stage of egg meiosis will take place after the sperm's nuclear material, the male pronucleus, is already inside the egg cytoplasm. Germinal vesicle, primary oocyte, first metaphase, second metaphase, meiosis complete. Microvilli, vitelline envelope, cortical granule, cell membrane, sea urchin egg surface. Cell membrane and extracellular envelope. The cell membrane enclosing the egg cytoplasm regulates the flow of specific ions during fertilization and must be capable of fusing with the sperm cell membrane. Outside of this egg cell membrane is an extracellular matrix that forms a fibrous mat around the egg and is often involved in sperm egg recognition. In invertebrates, this structure, usually called the vitelline envelope, contains several different glycoproteins. It is supplemented by extensions of membrane glycoproteins from the cell membrane and by proteinaceous posts that adhere the vitelline envelope to the cell membrane. The vitelline envelope is essential for the species-specific binding of sperm. Many types of eggs also have a layer of egg jelly outside the vitelline envelope. This glycoprotein meshwork can have numerous functions, but most commonly it is used to attract or activate the sperm. Lying immediately beneath the cell membrane of most eggs is a thin layer or about 5 μm of jelly-like cytoplasm called the cortex. It is stiffer than the internal cytoplasm and contains high concentrations of globular actin molecules. During fertilization, these actin molecules polymerize to form long cables of actin microfilaments. Microfilaments are necessary for cell division. They also extend the cell surface into small projections called microvilli, which may aid sperm entry into the cell. Also within the cortex are the cortical granules. These membrane-bound Golgi-derived structures contain proteolytic enzymes and are thus homologous to the acrosomal vesicles of the sperm. However, what is a sea urchin sperm contains just one acrosomal vesicle. Each sea urchin egg contains approximately 15,000 cortical granules. In addition to containing digestive enzymes, the cortical granule contain glycosaminoglycans, adhesive glycoproteins, and hyaline protein. As I will soon describe, the enzymes and glycosaminoglycans help prevent polyspermy. That is, they prevent additional sperm from entering the cell egg after the first sperm has entered. While hyaline and other adhesive glycoproteins that surround the early embryo provide physical support for cleavage stage blastomas. In mammalian eggs, the extracellular envelope is a separate, thick matrix called the zona pellucida. The mammalian egg is also surrounded by a layer of cells called the cumulus, which is made up of the ovarian follicular cells that were nurturing the egg at the time of its release from the ovary. So mammalian sperm have to get past these cells to fertilize the egg. The innermost layer of cumulus cells immediately adjacent to the zona pellucida is called the corona radiata. So cumulus, polar body, ovum, zona pellucida. Then next, how we recognize. Interaction of sperm and egg generally proceeds according to 5 steps. Chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. And binding of the sperm to the extracellular matrix of the egg or jelly or zona pellucida of the egg. First chemoattraction, then exocytosis of the sperm acrosomal vesicle and release of its enzymes. Passage through the extracellular matrix to the egg cell membrane. Fusion of the egg and sperm cell membranes. After the steps are accomplished, haploid gametes can meet and the reaction that initiate development can begin. So this is the next chapter. Sea urchins and other aspects of fertilization are dealt. First, we will be learning about the external fertilization in sea urchins. Sperm attraction, action at a distance. The events of sperm egg meeting and fusing are outlined. Like many marine organisms, sea urchins release their gametes into the environment. That environment may be as small as a tide pool or as large as ocean and is shared with other species that may shed their gametes at the same time. Such organisms are faced with two problems: how can sperm and egg meet in such dilute concentrations, and how can sperm be prevented from attempted to fertilized by another species. In addition to the production of enormous number of gametes, two major mechanisms have evolved to solve this problem: species-specific sperm attraction and species-specific sperm activation. Sperm contacts jelly layer, then centrioles are there, actin is there, jelly layer extracellular coat, vitelline envelope extracellular matrix, egg cell membrane, acrosome reaction, digestion of jelly layer, binding to vitelline envelope, fusion of acrosomal process membrane and egg membrane. So summary of events leading to the fusion of gametes in sea urchin. So which is external. The sperm is chemotactically attracted to and activated by the egg. Contact with the egg jelly triggers the acrosome reaction, allowing the acrosomal process to form and release proteolytic enzymes. The sperm adheres to the ethylene envelope and lies a hole in it. The sperm adheres to the egg cell membrane and fuses with it. The sperm pronucleus can now enter the egg cytoplasm. Species-specific sperm attraction has been documented in numerous species including cnidarians, mollusks, echinoderms, amphibians, and urochordates. In many species, sperms are attracted towards eggs of their species by chemotaxis, that is, by following a gradient of a chemical secreted by the egg. These oocytes control not only the type of sperm they attract, but also the time at which they attract them, releasing the chemotactic factor only after they reach maturation. In sea urchins, the chemotactic agents are small peptides called sperm activation peptides or SAP that diffuse away from the egg jelly into the surrounding seawater. One such SAP is ReSACT, a 14 amino acid peptide that has been isolated from the egg jelly of sea urchin. It has a profound effect at very low concentrations, such that the binding of even a single ReSACT molecule may be enough to provide direction for the sperm, which swim up a concentration gradient of this compound until they reach the egg. Then acrosomal membrane, pindin, sperm cell membrane, globular actin, nucleus, acrosomal enzyme, bindin, and actin, microfilaments. Acrosome reaction in sea urchin, sperm. The portion of acrosomal membrane lying directly beneath the sperm cell membrane fuses with the cell membrane to release the contents of the acrosomal vesicle. The actin molecule assemble to produce microfilaments, extending the acrosomal process outward. Acrosome reaction. A second interaction between sperm and egg jelly results in the acrosome reaction. In most marine invertebrates, this has two components: the fusion of the acrosomal vesicle with the sperm cell membrane and exocytosis that results in the release of contents of the acrosomal vesicle, and the extension of the cellular protrusion called the acrosomal process. The acrosome reaction in sea urchin is initiated by contact of sperm with the egg jelly. The protein digested enzymes released from the acrosome digest a path through the jelly coat to the egg cell surface. Once the sperm reaches the egg surface, the acrosomal process adheres to the vitelline envelope and tethers the sperm to the egg. It is possible that large complex of acrosomal protein digesting enzymes coat the acrosomal process, allowing it to digest the vitelline envelope at the point of attachment and proceed towards the egg cell membrane.